Hey everyone, we're back, still in our biochemistry mode. Now we're going to look at proteins and enzymes. We've already seen that enzymes would be important for breaking things down in our carbohydrates chapter. So let's jump on in and check this out. Proteins. The Greek word proteos means of first importance. Protein compounds are fundamental building blocks of many of the body's structures, such as nervous tissue, muscles, cartilage, tendons, and ligaments. Protein compounds can be formed in plant life by the addition of nitrogen from the soil to the carbohydrate compounds which were formed by photosynthesis. Protein molecules are relatively large and complex structures containing many elements and atoms. The words protein and amino acid become synonymous terms once it's understood how they are related. A protein is actually a chain of amino acid molecules linked together by a chemical phenomenon known as a peptide bond. The essence of a peptide bond is the alternating pattern of two hydrogen atoms and one oxygen atom on the external bonds of a protein molecule. So basically, amino acids are the building blocks of proteins. The smallest identifiable protein molecule is a single amino acid molecule called glycine. Glycine consists of a molecule of methylamine joined chemically to a molecule of carboxylic acid. Proteins are in a liquid state at body temperature, and protein is naturally hygroscopic and utilizes imbibition to maintain its liquid state. Due to its hygroscopic nature, proteins are considered to be the body's natural humectant and are essential in maintaining proper body moisture levels. Proteins are amphoteric, consisting of both acid and base parts. Because it is amphoteric, this aids in the retention of moisture by the body, even in excessive heat, such as fevers or hot weather. When the peptide bonds of a protein molecule are removed, water is shed by the molecule. Remember, it's tied together by two molecules of hydrogen and one molecule of oxygen. And that's water. The viscosity of the molecule increases as the water is shed. It loses water, it gets thicker. When all of the water is removed, a solid particulate called a coagulate remains. Coagulated protein is resistant to decomposition by hydrolysis. Formaldehyde can also remove the peptide bonds from a protein molecule. This is the chemical explanation for the drying out of body tissues which are exposed to formalin solutions. The stronger the concentration of the formaldehyde, the more moisture extracted, and hence the more firm the body tissue. Protein molecules can undergo decomposition by the presence of water, which causes a process called hydrolysis. Now, we've seen hydrolysis in other chapters, and as a friendly reminder, a number of chapters ago I said that books like this are written by different editors. Okay, some chapters might be completely written by someone other than the main author. Could be that the main author provided a framework and someone else kind of fleshed it out, which is why we see something that's like a process called hydrolysis. Well, we've seen hydrolysis a lot up to this point. Decomposition of a protein can occur in two ways. Deamination or deaminization, the removal of the, guess what, amine from an amino acid by another substance, and decarboxylation, the removal of the, you guessed it, carboxylic acid from an amino acid by another substance. It's right in the name. These are not hard to remember. Complex proteins can undergo partial decomposition. Instead of going all the way, they can just kind of break down a little. Partial decomposition results in the separation of the single protein molecule into the specific number of amino acid molecules of which it was composed and held together by peptide bonds. Complex proteins and amino acids can then also undergo complete decomposition. This results in the creation of carbon dioxide, water, and urea from the protein or amino acid molecule. Some energy is also released by complete decomposition. An organic enzyme is a compound which normally acts as a catalyst for some type of chemical reaction such as hydrolysis. A catalyst will alter the rate of speed of the chemical reaction to which it is introduced, but does not become part of the products of the reaction nor will it be altered by the chemical reaction. And enzymes are said to be substrate specific. So if you're having a little bit of trouble with this, it's real simple. It starts it up, it instigates it, it gets it going, but it doesn't become part of the result. It's that guy that starts the fight and watches it. And it's not going to be altered by the chemical composition. So everything that happens and everything that changes, this isn't going to change. It's not even going to be involved. It just starts it up, kind of walks away. Specific enzymes only act upon specific substances. The square peg goes in the square hole. The circle goes into the circle hole. The square peg might also go into the diamond hole if it's just a square rotated. If an enzyme is categorized as a member of the protease family, it will only act on protein substances. It will not help break down carbohydrates or lipids. Enzymes possess physical and chemical properties similar to that of proteins. One significant difference can differentiate proteins from enzymes. Proteins are crystalloidal in nature. They're therefore capable of passing through semi-permeable membranes, whereas enzymes are colloidal in nature. They're incapable of passing through semi-permeable membranes. The names of many of the enzymes end in the suffix ase. Besides being substrate specific, enzymes can also be temperature and pH specific. They only operate in certain ranges or in certain pHs. Enzymes are known to be labile. They have the capability of regrouping and returning to original strength after working on a substrate over a period of time. So basically, they do their job, they peter out, they recharge, and then they can go all over again. Because of this labile property, enzymes do not cease to act upon the death of the organism which created them. If there is no more substrate to act upon at the time of somatic death, the enzyme will then act upon the very tissues which created it, and this is called autolysis or autolysis. It will automatically attack the body's own tissue when it doesn't have anything else to attack. Your body will literally break down itself. Even the enzymes like lysozyme within each body cell will remain active after cellular death. It is essential that the exoenzymes and endoenzymes be neutralized after death to minimize the damage caused by autolysis. The cavities of the torso injected with formaldehyde-based fluid, which is cavity fluid, to neutralize the exoenzymes of the digestive system, and the arterial fluid acts upon the enzymes within the cells themselves. Remember, it destroys, it breaks down the formaldehyde breaks down the links within the protein. It breaks down the links in bacteria, and it also inactivates the enzyme so that they don't do their job either. So it's kind of the best trifecta ever for preservation. Folks, thank you for your attention. See you next chapter.